protein 60 is an important heat shock protein which helps anadromous fish to adapt in different environment. So in our study, we have conducted different bioinformatics strategy and experimental strategy to see how heat shock 60 helps Hilsha Shad to challenge the temperature stress during their migration. So to do this, at first, if you go to the methodology, at first, Hilsha's whole genome is sequenced in 2018, and up to date in the NCBI, only the genome is submitted. But there is still we don't found any gene that is separately characterized. So therefore, we have to predict the H60. So we take CRISPR Institute back. It is an anadromous model fish. We receive its sequence, the complete sequence from ancestry, and then we go through the genome blast with the complete sequence of Hilsha Shad. And after this whole genome blast, we predicted three copies of similar types of sequences like this or back throughout the genome of Hilsha Shad. And this is the table. This is the three similar sequences like this or back Bishop 360, and all of their identity percentage is around 80%, and their e-value is very low. That means we can assume that they are quite similar. So this is the bioinformatics part that we have conducted. But as it's a computational part, we have to confirm that either is it valid or is it exactly available or not. So therefore, we designed the primer against these three sequences, and we run the PCR, and we get our band exactly the desired location, that is below 1300 each amplicon size. We get the desired band in that range. They confirm that yes, these three sequences present in Hilsha Shad. Then we go to the expression that we need to. That's the key part of our study. And for the expression, we collected samples from three habitats. We collected samples from Podda. There is the fresh water. Then we go to the Vola, which is the brackish habitat, and then we go to the Kuakata, that is the marine habitat. So our objective is to check in these three habitats how this H60 modulated. And actually, how the idea comes, because according to the meteorological study, we have seen that in February we collected our sample this year, mid February, 16 to 18 this time this year. So in mid February, it's also known as late winter. So late winter is a weather transition period when winter closes and spring starts. So in this time, the water temperature of Podda is 27 degrees Celsius, and the temperature of brackish or estuary is 29, and in the same time the water temperature of Bangopsagar or Bay of Bengal is 26 degrees Celsius. So this is three temperatures. Hilsha, this is strategy. I mean, this experiment was done in February, which is the period of the downstream migration. I mean, in this period, the Jatka travels to the polar region, and then develops into intermediate phase, and then this intermediate phase further travels to more downstream to the Bay of Bengal. So this is the study was conducted in the downstream migration there. So we want to see how this gene responds in this temperature modulation. So to conduct that, we have collected samples from, I mean, RNA extracted from gill, liver, their muscle, and their kidney for different tissues. Then we conducted their cDNA synthesis, and we will run the qPCR again, very specific considered primer. And we have checked that liver and gill, liver and kidney showed the very higher expression. And in all three habitats, I mean, in all four of our tissues, the expression was very high in brackish conditions. If you see the kidney, the brackish is very high. If you see the liver, here also the brackish is very high. The muscle, here also brackish is very high. And for gill, brackish is high. All three genes, they show higher expression in brackish condition. That means from 27 to 29 degrees, this temperature 2 degree uprise causes their elevated expression, and when they go to the 26, they started to down regulation. So that means this up and down regulation suggested us their active function against the temperature modulation. Now the findings, I mean, second finding is why the difference is showing that liver shows higher expression than muscle, kidney shows higher expression than gill. Then actually this phenomenon suggested that during the migration of Hilsha, the liver faces most a higher temperature challenge. Therefore, the function of protein denaturation, I mean protein misfolding. Actually, Hilsha 60 expresses to prevent misfolded protein. They helps protein from misfolding and to keep their structure constant. That means in a tissue, if we found higher expression of Hilsha 60, it means that in that tissue the level of protein misfolding chance is high. So that means in liver, due to this temperature change, the misfolding of different protein is becoming increasing, and to suppress this, to prevent this, H60 is increased, shows increased expression in liver. In case of kidney also, because kidney's main role is the detoxification, and it helps to maintain the osmoregulation. And in case of osmoregulation also, H60 has the role, because to perform the osmoregulation, there are many ion pumps that are available in the kidney. So due to temperature elevation or temperature resistance, these ion pumps may rupture. So to prevent this ion pumps, HO60 helps. HO60 binds to these ion pumps and saves there as a role of savior. In case of muscle and gill, we found reduced expression. One question normally comes that okay, gill and the muscle continuously face or directly face the water temperature, but why their reduction is reduced? The reason is in many other anadromous fishes, especially in salmon and also in rainbow trout, it has been noticed that muscles produce different reactive oxygen species. I mean, superoxide dismutase, especially superoxide dismutase during temperature or any environmental stress. And to mitigate this, they also produce different kinds of enzymes as well that can neutralize this reactive oxygen species. And the expression of Hilsha 60 and the production of reactive oxygen species are interlinked in a feedback loop manner. That means if Hilsha 60 expressed in a tissue, it doesn't mean that it will neutralize the ROS, but it indicates that there may be the presence of ROS. So in muscles, we can assume that there are other strategies that is available to mitigate the ROS. Therefore, the expression is there is expression, but the requirement is not sufficient. That means there are other mechanisms is available in the tissues, in the muscles. In case of gill, we found expression, but not like the liver uh, kidney. The reason is gill has different physiological changes. It can change its physiology to maintain the temperature. Like it can remodel the lamella. Fish gill remodel the lamella, and they can run other strategies to manage the heat. Therefore, the requirement of H 60 is low in gill, because gill itself can take part in gas exchange role. So therefore, it has the capability of maintaining heat by different physiological activities. So from this overview, we can assume that in this downstream migration, the internal organs are facing more temperature challenge. And another finding is that in these three habitat, we can see that the expression of H 60 is high in the brackish conditions. That means brackish Hilsha's has better stress response capability. So, in a nutshell, what actually our study helps for the global, I mean, for the national perspective, because our country is trying to develop different sustainable aquaculture techniques for different fishes, and for hillside is also under planning or under experiment by different research groups. So, in this case, we can propose that from based on these three habitat, the brackish zone is more suitable to develop the sustainable aquaculture industry of Hilsha, because here naturally the fishes is getting more stress response capability.